Okay. All right. Thanks everyone for coming. Yeah, it's a bit of a, we had a lot of field trips planned, so it's really nice. I mean, yeah, it's a bummer, but it's good we can still catch up on Zoom and do these talks instead. So yeah, it was the other week I was out at my local reserve and noticed some wallaby grass, wallaby grasses with ripe seeds, and usually they're, their main time is sort of the November, December in the summer, but a few of them were still had ripe seeds. So I thought we could have a look at them and ID some together. So wallaby grasses are kind of one of the three big genera of grasses in southern Australia. You've got your kangaroo grass, your ostrostipes, the spear grasses, and your wallaby grasses. And you'll find them in a lot of different ecosystems, but especially your grasslands, your dry grassy forests, also up in the Alps. Yeah, widespread. So yeah, here I've pitched these pictures from my naturalist. We've got a real domination of wallaby grass on the right, and on the left, all these big tussocks are the red anther wallaby grass, which you'll find it in, if you go to grassy dry forests, you'll definitely see that species. So just a overview of Rytidosperma. It's really abundant in the southern half of Australia, but it's not restricted to Australia. So it's all over New Zealand, you can see. It's also native to Papua New Guinea, and there's some native species in South America as well. So I wanted to show you, I'm a big fan of iNaturalist. If you haven't heard of it, it's an open source website and a phone app for recording species. And it's linked to the Atlas of Living Australia. So if you
are we meant to sit iNaturalist?
Oh, whoops.
That's right.
One second. Can you see that now?
Yeah, that's better.
Okay, great. Yeah, so iNat is really taking off. A lot of people use it worldwide, and it's linked to Atlas of Living Australia, which is the resource that all of Australia's biodiversity atlas. I just wanted to show you, I like the taxonomy breakdown in iNaturalist. On Vic Flora, you can see, your kingdom, class, family, order, genus, species, but iNat breaks it down into some of the other subclasses. So you've got your subfamily and your tribe of grasses as well, which can be interesting when you get more of an overview of the different grass groups. Also wanted to show you just some of the records across the world of Rytidosperma. So heavily in Australia and New Zealand. Interestingly, no records in Papua New Guinea. But we can see some of these native records in South America. So you can, click on these, you can have a look. And I just think that's pretty cool that you can see that's definitely a Rytidosperma. It's got those fluffy seeds. Yeah. All right. Back to the presentation. So iNat's also great to just have a browse for different species, and you get heaps and heaps of photos because you have to upload a photo with your observation. So you can get a bit of a feel for what the different species actually look like if they've been ID'd properly, hopefully. Okay, so getting to IDing grasses, obviously they're different to our flowering plants with, showy petals. They don't have scents, they don't have colours because they're wind pollinated and they don't need pollinators. So that causes a whole bunch of different structures that, aid in spreading seeds. So with all those different structures, we've got a whole bunch of different terms. So a few different places to go for terminology. Is the Wallaby Grass website. Have a look. Yep. So this page has just a good overview of some of the different terms that are used and a bit of description and a bit more in depth about them. So this is a good website, as well as I'm sure you're all aware of the Vic Flora glossary. It's just up here in the Vic Flora website. This is great to, it's got almost all the terms that are in their keys. And the terminology can be overwhelming when you start, especially if you, sort of don't know every third term, but just Google everything and it will eventually stick. I still have to look up, lots of terms when I'm keying grasses. All right, so I just wanted to go through the different components of a grass. I'll break down some of the terms and things that are unique to wallaby grasses as well. So starting with the roots, there's three main types of roots. You can have rhizomes, so they're underground runners, stolons, which are overground runners. So your kikuyu might have this, and a lot of those lawn forming grasses, the weedy ones. But majority of Australian native grasses are going to be the tufted or the tussock forming, and all Rytidospermas are tufted. So you're not going to get questions about that in the key for Rytidosperma, but with the general Poaceae key, there might be some questions about the roots in there. All right, so the next important bit, and I'm just going over all of these terms, assuming no one knows anything about grass. So there's it took me a while to understand that the leaf is not just the blade bit, it's also the sheath. So that joins, that leaf generally starts at the node, the knobbly bit, and it wraps around the stalk, which is called the culm. It's in a grass, generally not fused, the sheath. It's generally split. If it's fused, you're likely looking not at a grass. And then there's always a distinct kind of junction between the sheath and the blade. So other graminoids like your sedges and that, and rushes can have sheaths and blades, but they won't usually have a distinct junction. Okay. Now, a unique thing about wallaby grasses, I mean, some other grasses can have this, but all wallaby grasses will have it, is their ligule is modified to be hairs on the edge of that junction. So you can see it, it's zoomed out a bit, but you can see it quite clearly on that grass there. They're all sticking out like a little tutu sort of thing. That's a really good character to know that you're looking at a wallaby grass if you don't have inflorescence. That's just another, really hairy ligule. Another thing to be aware of is the blade can be an important factor when you're IDing grasses, but it's generally not critical for wallaby grass ID because they can be hugely variable. So you can have very thin or very wide leaf blades within the same species. But something to be aware of, you might have tightly rolled leaf blades, or you might have flat leaf blades. So they can be quite distinct, but yeah, generally not critical. So the inflorescence in a grass is the bit that has all the flowers and all the seeds, the grass head. So the wallaby grass here, the kangaroo grass, and this is a weedy species. So that includes the whole head. It's called the inflorescence. So there's a lot going on in this diagram. This is just a schematic of grass structure and a few important terms. And these are fairly unique to grasses. So we might start with the floret. And so that's your kind of individual flower unit if you're thinking about plants. So these don't have the anthers and the styles drawn onto them, but that would be inside there. The caryopus is just the seed, fancy word for the seed. And the seed is surrounded by two husks, and these are called the lemma and the palea. Palea is just a small one, the lemma's the one that is often larger, and it will kind of sit around the palea. So generally all grasses will have that as the kind of smallest unit, and then you might have several of those stacked together, and they will sit inside two other husks, which are called the glooms. In this diagram, they look basically the same, which is not very helpful, but the glooms are sterile, so they've got no flowers, seeds in them, and they're just empty husks. empty husks that sit at the base and surround the florets, and they're basically protection while the florets, spread their pollen, and so all of that together is called a spikelet. So these are some wallaby grass spikelets. So you've got all your seeds sitting in those two big protective glooms. That's the glooms held back there, and all your multiple florets stacked together. So just a few more spikelets. That's a spikelet. That's a spikelet. All right. Now the floret doesn't look as simple as that diagram before, but essentially you've got the larger lemma and the smaller palea behind. The lemma starts out having those two lobes, these two lobes joined with the central awn, and then when it dries, that awn curls and does its thing, ends up like this. Okay, so have a look. Basically, all of wallaby grass IDing is done with the floret. So this is all you need, but it's not as simple, unfortunately, as just the lemma and the palea. All these other bits have names. So we'll go through them. So you've got your lemma body, which is the tip to where the awn starts to curl. Then you've got your lemma lobes, which are all of these bits, but the very thin bit of the lemma lobe where it contracts and it becomes very thin, that's called the bristle. You've also got your central awn, where the awn comes to the base, and there's that fork in the lobes. That's called the sinus. The bottom hard bit of the lemma body is called the callus, and you've got varying degrees of hairs. Sometimes they're in rows, sometimes they're all over it, sometimes it doesn't have any. So a useful way to remember some of these terms, Graham Lorimer, who's almost an authority on grasses in Victoria, he runs a really good grass workshop and has written his own wallaby grass keys, and the whole thing refers to the grass seed as a unicorn. So if you imagine some little eyes on there, this is the unicorn's face. Obviously the awn is the horn. The lemma lobes are the ears, and the callus is its snout, and the hairs are mustaches or whatever you want to call them. So yeah, that can be useful. So yeah, it's all about floret. The differences between the florets are obvious here. You've got one with hairs all over it, very short lemma lobes, very short awn, whereas the Rytidosperma, daunting, I've spelled that wrong, is very shiny surface, hasn't got hairs on it. It's got very distinct rows of hairs, huge long palea you can see pointing at the back, whereas this one you can't see it at all. Yeah, so it's all about observing those differences in the floret. Another important thing, when you find a wallaby grass and you want to ID it, grab the bottom seed, this will be the largest one, and you want to work off the largest one. These ones up here are tiny. There's some species that have very, very small seeds, and you're likely to be led astray by those small seeds up the top. So grab the bottom one that's closest to the glooms. All right. I was going to use a microscope to show these, but it's a bit difficult working through the key and sharing my screen. So I just grabbed some screen grabs of the Vic Flora key, and I thought we could go through a couple of species that I found this week and that I had photos of. If anyone has any questions about terms, if I'm not going over them enough, just shout out. All right, so first question, lemma with hairs in distinct rows, although these sometimes incomplete, otherwise glabrous, or occasionally with scattered hairs between the rows, or lemma glabrous except for marginal tufts of hairs. So the rows of hairs, the hairs at the tip are often called the callus tuft or callus hairs. They're kind of excluded from the rows of hairs. So the rows of hairs are going to be just above the callus, if there's one. And here, often they're called a row of hairs below the sinus, which is that point where your awn rolls down to. So this species, this seed, has that distinct row of hairs above the callus. It's got marginal tufts here, but that's okay for that step. The alternative is it's got hairs all over it. Yeah. Okay, so step 2. Lemma with a complete row of hairs below the sinus. So no, we don't have that. So we go to step 17. Also, I'd recommend when you're doing wallaby grasses, hop into the bracketed key. Just have a look at that. And if you're not sure, open up the options, have a look at the distribution. If there's no overlap in the distribution, it won't be your species, very likely. It might be, but so yeah, you can just open up your species in a new tab. You can check, okay, that one's very rare scattered distribution. We're looking at something, down in the Wilson's Prom. It's not going to be that species, sort of thing. I find that very useful. Okay, so next step. So, oops. We didn't have the row of hairs, complete row of hairs below the sinus. So next question, lateral lobes of lemma shorter than the body. So the lateral lobe is from the sinus, and this is my interpretation of it. It's quite difficult to find exactly where the measurements are supposed to be from. My interpretation of this is from the sinus all the way to the tip of the bristle is the lateral lobe. So shorter than the body of the lemma. And this is a grass of alpine bogs. So the bristle kind of disappears a bit there, you can't quite see it, but this is longer than from here to here. And that's your measurement for your lemma body, is the sinus to the callus. So we ruled out that one. Go to step 19. Hairs of lemma reduced to marginal and callus tufts, leaves rigid, usually strongly reflexed, and the sheath blade junctions, alps and subalps. So obviously when you read through the whole thing, I got this from in my 5Ks in Melbourne. It's not going to be alps and subalps, but it has this row above the callus, so it's step 20. Oops. Callus of all but the lowest florets in a spikelet elongated, 1.5 to 2 mil long. So this measurement, I believe, is from below the line, this row of hairs to the tip. That's also quite, I haven't been able to find an exact where they measure that length from of the callus. But that's my interpretation of it. So you can see here, these are 1 mil increments. So it's just on the 1.5 mil. The rest of this explanation, the upper florets far exceeding the glooms as a consequence, and the lateral lobes of the lemma contracted rather suddenly into the setae. The setae is another word for the bristle. You can kind of see that here in this top photo. They're rather broad and they squeeze in rather prominently. Some of these terms can be difficult to know, what is contracted rather suddenly if you're only, you're looking at a seed for the first time. Once you look at a few different species, you'll get a bit of an idea of, what's a gradual, gradual contraction into the setae, or what's a abrupt one. So if the callus was very small, I think this one's long enough. So this species is Rytidosperma racemosum. This species has very tight florets, so they're not spreading out everywhere like some Rytidospermas will. They're very tight. It's got the pointy callus. The lateral lobes narrow very quickly, and this species is very widespread across Melbourne. This one's, I've seen lots of florets out at the moment. And yeah, it's a hardy species. You'll see it in people's lawns, in bush reserves, everywhere. So this is a great one to keep an eye out for. It's a good one to start with. And it's got a very, distinctive inflorescence. It's very narrow. So once you get your eye in for it, you'll know it. I might actually just skip over this one because this species is very similar to Rytidosperma racemosum, and it follows almost the same steps. It just goes one more. We can come back to it if people are interested. So just to show a different kind of seed, this one has, and this is another one I collected this week, has hairs all over it. So straight away you can see it's quite different. So in the key, step 1, it doesn't have lemma hairs in distinct transverse rows, even though there's, there's kind of longer hairs at the top, a little bit of hair in the middle, and another sort of row. It's got some covering over the face of the unicorn. So that takes us to step 22. So there's not that many species with hairs all over them. So that's nice. It narrows it down a bit. So next step, hairs of lemma, more or less uniform in length. Or, so these are definitely not, hairs of lemma elongating gradually to the base, from the base to the sinus, or the upper row of hairs distinctly longer. So they're distinctly longer. That's unlucky. Okay, next step. Hairs of lemma uniformly short, except for a conspicuously longer upper series. Lemma less than 3 mil long. So I had to measure this before. There's our 3. These are in mil increments. So lemma more than 3 mil long. So that's referring to, when they just say lemma, they're not meaning the body and the lemma lobes. They're just referring to the lemma body. So that's because there's no species that are only, with their lobes and their body are 3 mil long. So this is longer than 3 mil. And the hairs are quite long as well. So go to the next step, 27. Lateral lobes of lemma are no longer than the lemma body. So you could look at that and go, okay, they're about the same length, but remember, it's from the sinus. So this is longer than from the sinus down to the callus. So 28. Palea, so that's the bit behind, sits behind. Palea exceeding sinus of lemma by up to 2.5 mil, never reaching midway on the lateral lemma lobes. So it's clearest in this bottom image that, you've got your lateral lobes here, that palea is not reaching halfway. That would be up about here. So that takes us to 29. If you've got a hand lens, you can pull that palea back and have a look at the shape of it. All of these terms of obovate, obtuse, they're all on the Vic Flora glossary, and they have now diagrams and photos to go with those of what those shapes, they've got the description, but they've also got a picture to go with it, which is very useful. So the key description in this step is the palea exceeding the sinus by no more than 1 mil. We can clearly see that sinus even from looking front on. So it's longer than that. I didn't, unfortunately, take a photo with the measurement, but you measure from where that awn peels away up to the top. And that's that length. So step 30, this takes us to nearly the bottom of the whole key. And these two species are very closely related. So you might get a sample that's somewhere in between, and that's normal. So it'll be one of these two. But this sample that I've got a photo of, you can go off the colour of the awn. So base of central awn pale, twisted no more than twice. This is not, it's quite dark and it's twisted a lot of times. Although I'm actually looking at this photo and that's not so strongly twisted. These are not the same seed, by the way. So I might have made a mistake there. The size of the whole inflorescence is important. The length of the glooms. I don't have photos of those, unfortunately. So this one is not pale and it's tightly twisted. So that takes me to fulvum, which I believe this species is. I just missed one point in this step back. The Palea has a slight twist in it. You can see that in this this top photo has like a quarter twist sort of thing, and that's actually a really diagnostic feature of both Rytidosperma bipartitum and fulvum. If you see that, it's hairy, it's got the twist, you'll likely have either of those species. I meant to put a questions slide there, but yeah, that was quite a fast run through on some IDs. But yeah, I hope that clarified a few of these terms and where to measure from, because it's not always clear in the key. And yeah, if anyone has any questions, I did offer if people could bring samples if they wanted help. So yeah, open it up now.
Hey, I have a question about these pictures that we're looking at.
Yep.
Are they of immature little spikelets, flowers, or are they seeds?
Mature florets. So
These are
when
mature.
Yeah,
florets.
so there'll be the flower head will all be dried out, and the florets will be kind of dry and falling out of them, and that's the seed that you want to ID. I'll just go back to, oops, this photo here, the bottom one, that's still a bit green. So it's not, I mean, it's probably possible to ID them, but it's a lot harder because all of the hairs are kind of still stuck down and the awn hasn't, curled out. So you want it when it's like this top image, when they're, really dry.
Oh, handy. That's good to know. Thanks, Nina.
Yeah.
Nina, can I just follow up that question with another one? Because I'm absolutely 100% here to learn. I know that when you take a fresh one where it isn't all curled up and you just chuck it somewhere where it'll dry out, it will curl up and you'll get those curly characters. But do you think the hairs would change or expand as the fruit matures? Like, would those characters change as well? Would you do it from a fresh one if you let it dry out and it would be the same?
If it's, like this photo here where it hasn't curled out yet. This is actually, this is Rytidosperma nudiflorum, so that it doesn't have hairs on it. It has a couple of little tufts at the side.
Yeah.
But I think if it's like at that stage, it would be kind of fully grown and just waiting to dry
Yeah.
out. So I reckon you could put it in, you know, in the windowsill and give it a go. Yeah.
Yeah. Cool.
Going off Jacob's question, I guess, would the caryopsis or seed be like tucked under in the palea, like in the Rytidosperma seed there?
Yes,
Because it's pretty hard to see, hey?
It is, and it's tiny. And do I have a photo of? No, I don't think I had a photo of that. Yeah, it's tiny, and but if you pull back the palea on a ripe seed, you'll see the on a ripe floret, you'll see the tiny seed just sitting in there.
Cute.
Yeah. This species is Rytidosperma geniculatum, which is, you'll find it on like really dry, exposed areas through a lot of Victoria, and it's got these little geniculate means like a knee, and it's got these little kneed culms, and it's quite a small plant, and it's got small seeds as well. So that's another one to keep an eye out for. Got a photo of its seed as well. This is Rytidosperma geniculatum seed. It's got like, if you make like a metal sign with your hands, it's got like really splayed, you know, lobes, and it's got a tiny awn as well. So the bristle barely exceeds those lobes. So that's another good one to keep an eye out for. Yeah.
I've got a question from Lilith.
Oh, there's heaps in the chat, sorry.
Oh, that's all
right. Depends where you are. So the question was, are wallaby grasses more abundant than kangaroo grasses, or about the same? So if you're in a like a basalt grassland, typically kangaroo grass will be dominant, but you'll have wallaby grass there. But yeah, if you're like in the grassy dry forest that I showed a photo of, you know, your dominant species in some patches might be the red anther wallaby grass. So depends where you are, but there's certainly more species of wallaby grass because there's only one of Themata.
Thank you.
And a good another tip with wallaby grass is just keep in the back of your mind where you are in the landscape and the sort of the ecology, the geology. Is it a damp spot or is it a dry rise? Because you'll often have multiple species, you know, co-occurring. But if you're in a wet depression, say, you might have this golden seeded one, Duntonian. They like, you know, your wet low-lying areas. But yeah, if you're up on a dry little exposed area in the bush, you might have this species. So yeah, noting those things.
This was really good. Super helpful explaining, you know, what to measure and the nomenclature, because that is a nightmare to work with if you don't do this often. I was, and your photos are amazing. I was just thinking, if you've got enough of those, maybe you put together some sort of visual field guide to
Yeah.
to wallaby grasses with those things, and maybe like little scale bars to help with
Yeah.
measuring the exact things.
Yeah.
Because I think that would help a lot, to have like a little cheat sheet, with good resolution pictures like this, because otherwise with the keys, even if you know what you're doing, it's really tedious, this will be much quicker.
Yeah, for sure. And there's species that you definitely, I mean, I've only the ones that I've shown in presentation, like I've come across. There's obviously a lot more, but we could definitely put together something like that, I think. Yeah. And
Yeah,
I'll share.
I've got a few growing in my front yard now, things that I planted last year from big and whatnot. So when they drop seed, I'll collect everything and assemble something like that.
Wonderful. Yeah, that's another thing that wallaby grasses are dead easy to grow as well. So if you have a parkside planting at your house, grab some seeds and sprinkle them around.
They are, because they survive in my yard. John and I were talking pre-COVID, Nina, about running a little short course on this sort of stuff. So I think you're hired for when we actually get to do that. I will pay you for your amazing photos. They're fantastic.
Yeah, I was actually got a new point and shoot camera, and my windowsill had the best light yesterday, so that's where this photo is taken. But the dino light's pretty all right as well. But it does fuzz out on the thin bits. And with workshops as well, if people have particular, you want to have a look at particular things, let us know. Or if you want to run one yourself, that would be awesome. Because got a lot of free time with these lockdowns.
Yeah, we should do one for sedges, because they're also really difficult. It's been on my mind for so long, to actually go around and collect them and do them properly.
Yeah, I'm not very good at sedges, so maybe you can run that one, Stan.
Oh, Stan, I need it.
Yeah, let's do it. We did one for Juncus the other week. Haley did that one.
Oh, did
I
you?
missed it.
I've
so
got
I
a few wetlands near where I live, so I can collect some and see, but it may be another couple of months until they start flying. So
yeah, that'd be awesome.
But when they do,
you can collect up bits as you go, and then sort of take photos, and then do stuff like this. I've definitely seen a few things out at the moment, bulbous genuses and other, I can't remember their names. But
genus of Pogren is like probably the most common one. So it's usually what you see is that one. There's a few that can be really tricky to tell.
Genus of Bogan. Got some growing in my yard.
Have you been around long enough to remember that, Iris from Netamark?
Pardon? Yeah, it was Netamark.
That was Sue's, I think.
Yeah, that was one of the first names I remembered. Yeah. And Chemist Killer. I can't remember the proper name now. Yeah. Anyway. Thanks, Nina. That was really great. I definitely learned heaps, especially about the callus. Didn't know that stuff.
The bit that sticks in the bit that sticks in your socks. That's the callus.
Sticks in your socks.
Yeah, well, that's how it gets in. That's the spiky bit.
Yeah, nice. There you go.
Yeah.
I won't forget that. And I won't forget the unicorn one. That
Yeah.
was pretty good.
Yeah, so I'll share this or put this up on our communal Dropbox that everyone can access through Facebook, and maybe on our website as well, the LTSA one, for people that are not on Facebook. And yeah, we'll do another one soon.
That's awesome. Maybe would you mind if we shared it when John gives his grass lecture next week? Just as a supplementary thing? That'd be amazing. Because we're not doing a grass prac this year. So
yeah.
Yeah,
The slides by themselves are probably not all that informative, but I'll share this video and people can watch along and look at it or whatever.
That'd be really amazing. We've got Jacob and Lilith here. But
getting a head start.
Yeah. That's awesome. That was so good.
No worries.
Sorry,
were you talking about me or to me, Alison?
What? No, about you, Jacob. Don't worry.
Cool.
So.
Just got news that my rat just got found outside, out in the backyard, and I was having a momentary heart attack. Yeah, very naughty. So, but all's well. He's back inside. So.
Is this a pet rat?
Yeah.
Okay.
Yeah, no, I'm not friends with the wild.